For that informational talk on investment. I would like to spend the next 10 minutes investing on talking about genome editing. So, imagine a world without malaria or without HIV, and imagine a world with plants that can grow in harsh environments, that can grow in even the most desolate places on this earth. All of this is possible with genome editing. You've probably seen pictures of her spread throughout campus, maybe it was in banners or posters, and it's only a matter of time before she gets the Nobel Prize. Her name is Jennifer Doudna, a molecular biologist at her very own UC Berkeley campus, who is accredited to having discovered the mechanism for genome editing. Ever since her discovery, there has been mass press on genome editing. Magazines like Time, Nature, The Economist, and Wired are all mainstream news sources that have put the topic of genome editing on the front cover of their magazines. So you hear all these words like CRISPR-Cas9, DNA, RNA, sequences, and you see all these buzzwords being thrown around, but what do these actually mean? And so today I would like to sort of piece together the basic molecular bio techniques and fundamentals so that we can kind of become more familiar with the terms on this board. So let's begin with the fundamental yet essential flow of genetic information. First, we have DNA, and DNA you can think of like an instruction manual. And from this instruction manual produces RNA. And if DNA is the lab manual, then RNA you can think of as a copy of a specific page within that lab manual. So different RNAs will be different pages of the DNA, and each RNA will encode a different protein. And proteins are really just basic building blocks of the cell in the body, and you can think of proteins as the basic functioning unit. And so when someone says they're lactose intolerant, that just means they lack the enzyme, or lack the protein called lactase. So let's go a little bit more in depth into what DNA actually is. DNA is composed of 4 nitrogenous bases, which you can really just think of as Lego blocks. And I really like this picture because it's color-coded nicely, so that it has green pairing up with the red Lego block, and the purple pairing up with the blue Lego block. And the green cannot pair with the blue, or the purple cannot pair with the red. So it's highly specific. And this the DNA is actually double-stranded, meaning it has 2 strands. And so because of this highly structured pattern of Lego blocks, if you know the exact order and the color of the Lego blocks on one strand, then you can know the exact color and Lego blocks of the other strand because they match. Okay, so I told you that RNA is a copy of the lab manual, but what do I really mean by that? Well, shown in green is a little machine that, depending on the cell signal, sort of like what Nick was talking about actually with all those cell signaling, depending on the cell signal and anything like that, the green machine will know exactly where in the DNA to go to. And once it does so, since DNA is double-stranded, it'll sort of unzip the 2 strands apart, sort of like a zipper on your jacket, and it'll start to place Lego blocks onto one of those strands. And like I said before, specific Lego blocks correspond to specific other Lego blocks, and so it has a very highly structured sequence. And so what's pictured here in red is the RNA, and eventually this RNA now will be read by another machine that's gray in this picture, and it'll sort of just zip along this RNA strand and start to plop these little beads and start to form a strand of beads. And so what are these beads? Well, they're amino acids, and there are 20 amino acids, and when the amino acids are strung together, they're called peptide. Well, remember how I said RNA and DNA have very, very valuable information, and that is simply because the Lego blocks correspond to specific amino acids. So again, the specific sequence and the color and the order of these beads correspond to the RNA. So they're highly, highly dependent on each other. And so once this once the machine goes all the way to the end and produces this nice strand of beads, then the peptide will do something really interesting and it'll start to fold. And once it's in this nice folded state, that's when it becomes functional. So just to recap, I know that was kind of heavy. First we start off with DNA, then that DNA opens up, and then a new machinery can start to plot different Lego blocks together, forming RNA. This RNA will then be read by another machinery, which will plot the corresponding beads or amino acids to produce the peptide, and then the peptide will fold naturally to produce protein. So, going back to genome editing, what exactly is CRISPR-Cas9? Well, it's just a protein. And it's a protein that has two molecular scissors that really just clip the DNA. And so since I mentioned that DNA is double-stranded, it has two scissors, and it creates this double-stranded break. And normally that seems very scary, but in reality this happens all the time. Every time you step out in the sun and are exposed to UV light, your DNA is constantly breaking. But fortunately, there's a machinery that'll close that gap right back up. But if you can imagine inserting your own DNA, your own desired Lego blocks into this gap before the machinery can close up, then now you have all this new information, all these Lego blocks in which you can form RNA and eventually protein that was never there before. And alternatively, you can even create another break here, if you cut here, and that you can remove the DNA segment here. So if there were any mutations here that were deleterious, you can remove that very, very effectively. And I want to mention the CRISPR-Cas9 is really, really specific in that it has something called guide RNA, and that really is just Lego blocks themselves. And so the Lego blocks will zip along your DNA and track where the Lego blocks exactly fit, and that is how it knows exactly where in the DNA to cut. Otherwise, it could cut anywhere. And the beauty of this machinery is that scientists can put their own guide RNA. They can put any guide RNA in there, and it'll cut exactly where you want it to. Okay, so with all of this technology, it opens doors that were never dreamed of being opened. Wouldn't you like your child to be resistant to deadly or debilitating diseases? Of course, and CRISPR-Cas9 can do that. You can also delete certain mutations that are harmful for Alzheimer's or stroke, or maybe even opioid resistance as well. But you would also want your child to maybe have decreased risk of blood pressure. Maybe the allergies are really annoying, so maybe we can be resistant to allergies too. I always wanted to be tall, so maybe my child can be a little bit taller. Blue eyes are really nice. Maybe blue eyes with 20/20 vision too. So you can quickly see how appealing this technology really can be, but it also raises a very interesting ethical question of where's the cutoff between editing the genome for functional and health purposes versus just something that's convenient and nice to have. And so with this technology rapidly advancing, also comes great responsibility that we really need to think about. But for now, don't go assuming that your friend was genetically engineered to look better looking than you or taller than you or anything like that. There's still a lot of research that needs to be done in order for this to hit the hospitals. And Jennifer Doudna once said, the more we know, the more we realize there is to know. And while that's absolutely true, we are learning so much and very, very fast. Thank you.